Great, thank you so much. I'm going to put a link into the chat to our interactive Padlet board. For those of you who have been on these conversations in the past, you're probably familiar with this interface. In addition to the Zoom chat, we'll be using this Padlet today for feedback and interactions with everyone who's on the call. So please feel free to put comments into that Padlet at any time, and we'll pull some of those threads into our discussion as we go. As Aditi said, this conversation is hosted by the Training and Education Working Group, of which I am one of the co-chairs. My name, obviously, is Christina Baer. I'm the director of the Light Microscopy Core at UMass Chan Medical School in Worcester, Massachusetts. And I've been the core director for about 8 years. We have a number of people on this call today. I'm really excited about our group who's joining us today for this discussion about image analysis. So as our light microscopy technologies in the cores continue to advance and our image analysis workflows get ever more sophisticated, all of us core facility directors and teams face an increasing pressure, right, to support users with a wide variety of experiences, both on the microscopes themselves and with the different software tools that are necessary to pull quantitative data right out of the images that are collected. So the goal for this panel today is to bring together experts from both academia and industry to talk about the intersection of light microscopy and image analysis, and to explore how core facilities can better prepare researchers for success when it comes to the specific analysis side of things. So I'm going to leave it to the panel to introduce themselves, and I'm going to pass it around to, I think we'll start with Caroline.
You are muted.
There we go.
So sorry. Well, there we go. Hi, everybody. My name is Caroline Miller. Some people also call me Mills. You can call me whatever you'd like, either Caroline or Mills. I am an image analysis specialist and a histologist, and I work in the Bay Area. I've been a histologist for over 20 years. I've seen clinical laboratories. I've worked in research labs. I've worked in core facilities. And now I support small to medium-sized biotech companies in the Bay Area. I help them with their histology procedures, and I tend to do a lot of image analysis with commercial software using Visiopharm and Halo. Okay.
Perfect. Daniel, did you want to chime in?
Sure. Hi, my name is Daniel Miranda. I've been in industry for about 15 years now. I started off at Zeiss, where I went to the launch of Zen in Germany, and then I worked for Bitplane and Amars for about 9 years as an application specialist, and now I've been here with Leica as the Avia specialist for about 5 years. So I've seen a lot of the 3D image analysis world and commercial software space as well.
Awesome. And I think we've got Nellie joining us too.
Yep. Hi, everybody. So I'm Nelly. So I'm an imaging scientist at the Advanced Bioimaging Facility at McGill. So I'm worked there. I started maybe nine years ago. So basically, the daily task is to train people to use our systems like all the microscope, but also after to switch for the image analysis parts. So we have multiple workstations that they can use with software like Imaris, for example, but they can also use more like open source software. So we have powerful workstations, but like that they can just use whatever they want, depending on the size of the data set they have required. So we try to have them from the beginning before they start to prepare samples and things like that, just to be sure that it will work through the image analysis part until the end, like having some like real data analyzed and so on. So yeah.
Awesome. Thank you. And then we've got Evgenia. I don't know if I'm saying that correctly.
Close enough.
Okay.
Hello, everyone. I'm Evgenia, and I'm assistant professor at the University of Colorado, where I work for last 15 years, I think. And I'm cell biologist, histologist, microscopist, and I do a lot of image analysis throughout whole my career. And I started with cells, imaging and analysis, and then added histology to it. And 5 years ago, in addition to my lab, I organized the core here at the University of Colorado at my section. And I'm helping here with the sample preparation, with the imaging, and with the whole slide analysis, in addition to cell analysis.
Wonderful. And do we have David with us too, I believe?
Yeah, so my name is David Kiechenbüchler. I came from biophysics. I joined this core facility 10 years ago as somebody who helping with the analyzing. And from then 10 years ago, it was a little different, but we grew this into the current stage for now and to helping in core facilities and include the analyzing in the workflow.
Awesome. Awesome. Thank you all. I'm really excited to have you all here today and to hear your different voices coming from the sample prep side, more of the image analysis side, and those who work at the intersection of both the microscope and the analysis side. So I'm going to pass around a couple quick fire questions before we kick off a more in-depth conversation. So what is the most common imaging mistake that you see that ruins downstream analysis?
Are we just talking?
Yep.
I would-
Go for it.
I would say tissue quality, really. For me, it's if you have really bad tissue quality going in, then you're not going to be able to get anything good coming out.
Garbage in, garbage out. I'd say we're all pretty familiar with that.
Yeah, it's the same from my experience. Like if you have bad sample preparation, it's doesn't matter what you do on the microscope after that. And when I wasn't having my core here, and we have 3 other cores for imaging here, and they don't have people who are helping with sample preparation, I was telling them for last 10 years that, hey guys, you need to have someone whom people can come and talk to. Otherwise, like they come, they get disappointed, and then they don't use your facility anymore.
For sure. That's definitely a challenge. And particularly people, I think, don't often understand the expertise that goes into producing good quality samples, right? And at a certain point, I put it on a slide, it'll be fine, which we all know is absolutely not the case. Nellie, do you see similar things in your role?
Yeah, for sure. And one of the things that I've seen a lot also is like people that start to image their samples, but they don't really think what they want to analyze and what they really are interested in the samples. So they don't really plan the labelings and all the things on it, like to be able to extract the information they want. So they just arrive with sometimes a really beautiful like data set, but with the labeling that is inside, we cannot really answer their question. So yeah. It's a big issue.
I think it really starts way back when you're planning your experiment, right?
Yeah, exactly.
So when you want to know whether you're doing the right type of tissue, the right kind of prep, frozen, paraffin, cells, whatever, but you need to actually have a good idea of how you're going to answer the question you want to get to. So start with the question and then work and find the resources to answer that question. Is how I like to run these experiments.
So one of the biggest challenge I had is really to have them like early enough in their projects just to be sure that they will go the good way and not coming with hundreds of pictures that are not like or not for what they want to see at least.
Absolutely. That experimental planning is so key. Danielle, Miranda, I'm curious how this translates into the support that you do at Leica for Avia.
Yeah, it's interesting. Avia is platform agnostic, so I see images from every scope very often. I think one of the things that I encounter that's frustrating and disheartening is someone that is fully committed to imaging with a tool that is what they have, but not what they need, for example. And sometimes it's a factor of geography or money or sometimes politics, which I try to not get into. It's really sad when I'm just like, your time resolution, you need a spinning disk, and they're like, we don't have a spinning disk at the core. I was like, but I know someone 2 floors down that has it, and they're like, oh, we can't use that one. I'm just like, oh, it's so depressing. That's usually one of the things that I find most frustrating is when they feel like they're forced into using a tool that maybe isn't optimal for what the question they're asking.
That's also another planning thing too, right? Like actually making sure you have the resources before you start the experiment, and reaching out to try and find them.
Mhm.
Absolutely. Absolutely. David, do you have a opinion about which analysis tool a beginner should learn first? Where do we tell people to start?
That's hard. So technically, the answer is go to freeware because any paid version is hard to get. And the freeware versions out there is the classical answer would be then the Fiji stuff. But it's the landscape gets more diffractive in now we have specialized good tools for analyzing big histo stuff where Fiji is not so great, or having multiple images in there that would be cell profiler. But my guess is the first tool would be something that is free and learn the basics of the one-to-ones, the what bit depth, pixel size, saturation, blah blah blah. And it doesn't matter which tool, but because they all do the same. But the histogram, I want that at least you are capable of opening an histogram and get an idea what is showing to you.
Yeah.
Yeah,
Yeah.
the histogram, I agree. That's the first tool. I've seen so many overexposed images that then you cannot analyze them. Hey, you need to go and retake them, unfortunately.
And well, one of my questions was going to be if you could ban one bad habit at the microscope, what would it be, right? That negatively impacts your analysis. And I think it sounds like we're coming to an agreement here that it's definitely both not understanding bit depth and not taking advantage of investigating your histogram and making sure that you're scaling your, intensity values appropriately, et cetera. So do people have a perspective on what the most overused analysis method is? I know I have one, and it would be co-localization because it's used so badly, so in so many different ways. That is my least favorite tool when people come to me and say, I'm going to do analysis and it's going to be co-localization. But what do the rest of you think?
So I can comment on this. When I talk to 10 people who want co-localization, 10 mean 10 different things. Because for a reason I don't understand, they're just making up something in their head where they say, I want whatever. And then at one point it's distance, then it's if it's positive or not. And the other thing is stuff. So co-localization is one of my stuff where the red flag goes up when people say, I want co-localization. I'm like, oh, we have to talk because this is normally not a good thing. But this relates to the question from the beginning. When what instrument to use, what NAS to use is, you need to have the knowledge from what instruments are available and what technique is up to date. So what I see often related to the first question is when somebody came to me and say, David, I want a 100x and confocal and co-localization. And then I know how old your PI is.
Yes, I agree with you. It's a messy field, and I feel like we haven't defined our terms well, right? And that's part of the user education perspective on it, in terms of people coming and asking for things that are either impossible to do with the quality of data that they have, or just don't make a lot of sense, right? And sometimes their PI is asking for it, and sometimes it's coming from the users themselves, and that's definitely a challenge in my core facility. Do people have thoughts on the most underused analysis method? Do you have a favorite that's not very popular?
I've got a controversial here for
Oh,
both
good.
most overused and most underused. I'm going to say deconvolution. It is very frequently overused in situations where it actually makes analysis harder, and is very frequently underused in situations where you would clearly benefit from just a little bit of deconvolution, whether it's what's on the scope, commercial, Aries scan, Lightning, or it's something after Huygens, some free deconvolution. There's so many avenues for you to take if you're looking to resolve like small things in a very like hazy image, versus if you're looking for a nice homogeneous signal. throwing it into deconvolution just creates this wonderful heterogeneous span of like little tiny dots of fluorophores that you have to then blur and smooth just to get something that's homogeneous again. So you're shooting yourself in the foot. So not knowing when to and when not to do decon, I think, is a over and underused thing.
So along those lines, do you have thoughts about how we teach users that, right? That comes out of experience and it comes out of seeing lots and lots of images, which we're doing of different types, right? Which we do in the cores. Is this a situation where the answer is teach the users to come to us for help? Or is it a situation where there's an effective way to teach it?
Well, I think it should be both,
Yeah.
because me and Caroline, we organized a course of lectures where we give the basics, but then outside of those lectures, people can still come and ask the specific questions, because you can't cover everything in the basic course, but you can help with like details they need to know. So it should be both.
Yeah, and actually getting them used to asking for help too, as well, like creating that kind of allowing to have that conversation, right? And making sure that your users know that they're not working in a silo, that we're here to sort of support them. And I think, I'm happy to pass on my brain knowledge to anybody who asks me, but you've got to get those people to ask the questions first too.
For sure. And I think we've got a question in the chat that Daniel and others may have thoughts on about, is there a favorite free deconvolution software? And maybe we can keep that discussion to the chat, I think, while we keep talking about more generalized image analysis topics. But feel free to chime in there for sure if everybody's got everybody.
Personally, I had in undergrad, I'll put it on my face, biomedical signal processing and signal modeling. This was part of my undergraduate curriculum, and
I love it.
you learn about everything from Fourier transform to deconvolution, and it was just one semester. Not everyone gets that. I was a biomedical engineer major, so it was more on the engineering side, but some aspect of that kind of education really early on is super useful for a lot of different avenues, actually, whether they want to go into, more computer science or anything like that. I think that's the time to learn about those things. It's like sort of a foundational science, but really, like you were talking about experiment planning. I love Microsoft Paint and any just whiteboard-esque application that you can
Yes.
use to just draw something out. And one thing that I frequently do is just list all the tools in a toolbox and try to figure out, maybe rank them. This is probably going to be super helpful. This might be a little helpful. This might be not helpful at all. And if you as an advisor or an instructor can help flesh out that list of tools and help sort of rank those tools, I think that can really help, because that just puts options on the table. And even if you put a tool there that you say this is not going to be helpful, that helps for the next experiment they're going to do. At least they know that it exists. When they might ask you the question, well, would it be helpful in this case? So I think
Absolutely.
often we just sort of silo it and be like, you just need to do this. You don't present the negative options. Then the next time they go to do something, they just use the same tools that they know about, and they don't know that the negative options might now be the best ones for the different applications. So.
Well, and that's part of the challenge, I think, that at least in my core, we find when it comes to supporting image analysis software, right? Because we don't necessarily have the time to teach the full breadth of the toolkit that is available in the different image packages, right? And so it becomes a question of how do you communicate that to your users? And I think it's a critical part of teaching image analysis that we can't necessarily do in my core just because of time, right? And support. And I think another challenge that I'm seeing a little bit on the Padlet and a little bit in the chat too is having the time to learn these toolboxes in depth is also a challenge too for core directors that are handling a number of users and a number of different demands on their time. So I think that's that's super challenging as well. Please do feel free to drop resources that anyone finds useful, including your textbook, Daniel, or the links to the lecture series that was mentioned or anything like that also in the chat as well for people to reference, because I know we're all eager for all of these resources too. There were some additional questions about deconvolution in the chat, and I think I'm tempted to do a separate conversation all about deconvolution, not to put Daniel on the spot to help us with that one too, but potentially in the future, if people are interested in having a topical conversation as one of these meetings about decon, we certainly can do that. So I wanted to ask too what skill gaps people most often see in users transitioning from the microscope operation to quantitative analysis. So like what tools are missing amongst the users, and are there approaches that you take to try to fill those gaps yourself? I don't know if I'm looking to see who else, if Nellie or any of the others have some thoughts about that. Sorry, go ahead, Caroline. Yeah, please.
Well, I sort of see it on different levels, right? I have people that sometimes they're like full-on histologists and we're just learning image analysis. Sometimes they're cell biologists. So really, I think you have to assess where the person's coming from and what kind of experience. I also work right in the middle of people who are complete computer scientists that know nothing about histology. So therefore, I'll have to sort of teach them backwards that way. But I have found that since I'm a histologist and I learned these tools as a histologist, not a computer scientist, I tend to sort of teach them that way. And I teach different types of techniques. Like I talk about the machine learning, or I'll talk about regular thresholding, right? And trying to then relate it back to the image itself. So making sure that that person understands the different components of the image and how we can analyze it separately. Because there are some basics like thresholding, machine learning, and AI, right? Sort of neural nets. Right. So what can you do with each one of those techniques? And if again, starting with the question and making sure that we should already have a set of tools in place before they've even started the experiment, so that we sort of have an idea. So then we can teach them and they can learn those processes as they're going through the experiment. But making sure, again, the planning part is really important to make sure they have the right tools. Is that okay? Yeah.
Absolutely. Absolutely. So what I'm seeing too in the Padlet is that there's a large proportion of the audience that are dependent upon Fiji mostly, right? So I was going to turn our conversation to, the intersection between open source tools and commercial platforms, right? And how do you decide which way to go if you have the luxury of deciding, right? Some of us don't have funds for licenses for different analysis platforms. And I know we all have a different suite of software that we have access to. But is there a balance between the vendor provided tools, open source software, and custom pipelines? Like, how do you thread that needle? Is that something when you're providing image analysis services, where do you go first, and what is the easiest to onboard users onto? I think David might have some thoughts about this.
Okay, so for me, the commercial and non-commercial side offers the same toolboxes. There are no tools that are only here and only here, but the commercial side are the polished ones. So when I have users that I have a low experience and want just something nice to click through, they are the commercial ones. And on the opposite side, when they have the experience, new student who's capable of computer scientists, I can even say, okay, go with Fiji. There are some nice tools there. Get your own AI stuff to work. So for me, the distinguishing where to go is fundamentally how easy or good the user is with stuff. For example, people in Imaris can do very fancy stuff in 3D with a very low knowledge because it's nice and click and blah, blah, blah. To do the same thing in Fiji, you can do this, takes a lot more of effort. So you get the same numbers, the toolboxes are the same, but the polishing between the Halo and Fiji is on a different level. That's the reason you pay the money. You pay the money for the polishing.
Yeah, just to add a little bit of that, I find as a histologist, again, not a computer scientist, what I find that I use Visiopharm, and I'm now trying to translate my skills back into Fiji and Python, but I learned the process in Visiopharm, and it's because, you can call the images, you can analyze the images, we can measure the images, and we can export the data all in one shot. Versus sometimes that gets a little hard when you're using the open source software and things get confused and a little convoluted. So yeah, I totally agree with you, David.
And we can relate this back a little bit to the question that is hanging in the air, the deconvolution. When the deconvolution gets easy and accessible and you don't have to type anything in, because, for example, we are Nikon Imaging Center, we offer this to all the users and everything is in the file. It's literally a 2-button thing to get a deconvolution. People are way more willing to do this when I show them a free tool. They're like, okay, this is the NA of the objective, this is the mounting media, then the whatever, and they're afraid of typing this in. So when there is just a 2-click, people do convolution all the time and try this out. When there's a hassle of at least 5 fields you have to fill out, the success is low. And 5 is enough.
I think the user interface with a number of these tools is so important, like we're talking about here too, right? And also the supporting documentation, right? So, I started doing image analysis way back in my postdoc longer ago than I want to admit. And one of the first tools that I really heavily delved into was Cell Profiler. And I find Cell Profiler to be quite well documented in terms of the tutorials and the examples that are out there. I'm curious though, on the commercial side, have people found those tools to have sufficient documentation, or are you still looking for additional reference data sets and examples and that sort of thing as you go about doing image analysis with your users?
Normally, the commercial side is very good in this because this is where they come from. So Imaris, for example, has tons of videos out there how to do stuff, and it's easy for the users, and I always point them in this direction. But we have to be aware of that at some time point you reach a level where the company don't tell you what they do. For example, I had this with the spline algorithm in Imaris. We fitted the spline to an actin filament. The reviewer asked about what function you use. We asked the company. Normally they're very nice, and at some time you reach level, no, that's a secret. So this is a part of the commercial software we have to be aware of. There are always part in there where you say, okay, split cells, most likely it's a watershed, but what tiny nitty-gritty they do, you don't know. You will never find this out. So when we use commercial software, the reproducibility around the world is very low because they literally have to get the same version of the same stuff on the other side of the world to see what you did. We just have to trust that they did it right.
Well, I think it sounds like that's also a place too where reference data sets can be so useful, right? And so if there's a data set that you can run the comparison in different software platforms, that might give you some, comparison at least to see what numbers you believe or, appear to be appropriate. There was a question from Hannah too in the chat about how you validate volumetric measurements on open source software. How do we know that our open source platforms are providing realistic measurements, right? You can't do everything in a commercial package that's completely validated and tested most of the time. So, how have you validated the platforms that you use? Are there red flags that you look for when you're doing image analysis? How do you approach ensuring that the tools are the right ones for the task? Anybody have thoughts there? Or do we not? We just go with it and say, well, it's a tool we use,
We
right?
can have another talk on bias and sample size and cherry picking and general scientific concepts, but I think the tool plays a small part in the overall expression of what you're talking about, because essentially any tool can be subject to this, right? Really. So when I get asked this question, how do I know this is right? We're looking at one image and sort of one iteration of the first pass of segmentation. I'm like, we don't know yet. This is not science. Science isn't do something once and make a conclusion, right? It is test and check and increase the sample size and vary it a little bit and see how it responds and et cetera, et cetera. And I think too often we want sort of the silver bullet because we're just done, or we have a timeline and we have a deadline and we're like, I already did all this testing and optimization and science in my sample prep and my staining protocol. I don't want to also have to do it in my analysis. And it's like, sorry,
Yeah,
you're
that's
not
what you
done yet.
get.
You,
Yeah.
you don't just get off the airplane and magically arrive at your hotel. No, you still
Yeah.
have to go through baggage claim and rent your car and there's still more work to be done, even once you have the image in terms of scientific rigor, right? So generally, any of these tools, whether they be free or commercial, you can apply scientific rigor. You can apply, bigger sample set. You can filter out, things at the top and the bottom of a confidence range, anything like that. So those sort of principles are universal, whether you're using commercial or freeware, I think.
And just one point on that, I'm in the process of doing some phase 1 clinical trials, and let me tell you, the validation process gets even higher when you get into that stage as well, right? So it can often depend on the type of experiment you're doing. I'm not saying that not everything should be a quick and dirty, but sometimes there are quick and dirty experiments to see what's going on, right? That then you investigate going forward, and then you have other types of experiments that are also that are going to go into human clinical trials. And in that case, where you have extreme rigor, where we are doing validation sets and manually annotating hundreds and hundreds of objects and then getting the software to test it for us and then doing a comparison of those analyses in an F1, F2 score, right? To actually see false positive, false negative rate. So I do feel like there is some level of validation that happens for some of these commercial softwares that happens at some CROs and maybe some core facilities as well, where they are doing just quick spot checks to make sure the software is doing what it says. But ultimately, remember, we have our eyeballs, right? Our eyeballs are really important for this because we are going to be looking at these images and making sure that the segmentation and the classification that we've done is actually what our eyeballs are seeing in these images. So actually having a touch of your data and making sure you understand the data set and making sure that you look at the segmentation throughout most of the data set, right? Don't just run this analysis and export the data and be done, right? You want to always be looking at the data and making sure that the analysis that you've done makes sense.
I just want to double down on something you said, the human eyeball, right? And this might be a good transition to some of the other questions that Christina had. But for any AI, quote unquote, deep learning model that exists out there, whether it be commercial or free, it was trained by human eyeballs. Whether it be full self-driving or whether it be something fancy in an MRI or a CT machine, it was trained by human eyeballs. And you're, I don't know if in the academic world they're exposed, but there's a whole industry of companies that will essentially sell you human eyeballs, and they're located in other parts of the globe. And you will basically meet with them remotely, teach them what it is you're looking to extract from an image, whether it be a tumor or a cell, alive, dead, a fire hydrant, whatever it is, and you pay them a big gob of money to hire hundreds of people, if not thousands, who will scour hundreds, if not thousands of these images and manually annotate everything to validate these models. So at the foundation of anything that you think is fancy is manual annotation. And even if you're doing something in a very small scale or medium scale, I think it's very important to know that you can compare an automated result to a manual segmentation result and use that difference to sort of validate your method. It's time consuming, yes, but it is like the gold standard for comparing whether something is good or not, whether it's pathology and you get a pathologist to score a data set on 1, 2, 3, 4, whether it has so much cancer, and then we take the AI model and we have it do an analysis, and like you always have some type of human comparison. It's okay to do that in a small scale, just crop a couple of regions and just test it yourself. And it's even okay to do it in a large scale if you really want a truly robust comparison of how something is going.
That popped up. I'm not sure
Yeah.
why we wanted that. Okay, perfect. Great. No, I think the conversation is going in fantastic places. So, what have we not talked about at this point when it comes to analysis? I can throw some more questions at you guys, but are there topics that you think you'd like to talk about here in terms of teaching analysis, favorite analysis tools? We talked a lot about planning, right? And how important planning is going into an experiment. But I know all of us are also struggling with the breadth of tools that are out there and finding time to learn how to use them. So any tips, suggestions, or tricks on that front?
I'd say that they all have different words. Sometimes they use different words. There's different words to say the same thing, and don't be put if the exact workflow that you want in one piece of software doesn't exist in another. Right? Just be open-minded to the way different developers have developed these tools. And there's always a reason why QuPath does something this way and Visiopharm does something this way, and sort of really understand the engine underneath, right? Versus before you get all freaked out and it's not doing the same thing.
Absolutely.
I
Go ahead.
was
Yeah.
asked the golden question, what are you looking to quantify? The imaging is, do you have the right pieces? And the analysis is, do you have the tools to put them together? If you don't have one or the other, then you can't complete the jigsaw puzzle, right? And if you don't have the picture of what the jigsaw puzzle is supposed to look like, then you're sort of in the dark, right? So that first question is, what's the goal? That's getting the picture of the puzzle. And then you have to figure out, do you have the pieces and do you know how to put them together? And those are sort of the two, the imaging and the analysis side of things. I think very often you try to think, okay, I have the picture and I have the pieces, and they're like, it should be easy enough to just shake the box and have them come together magically, right? And it's not quite that way, right?
As someone said on the Padlet, I'm a doctor, not a wizard. We're not necessarily going to be able to make everything happen from your data, right? And I love that. I think
Yeah.
that's fantastic. Yeah. Yes, I know that being asked to do image analysis on a data set you've never seen does often feel like having to be a wizard. So it's always interesting to see kind of how collaborations to help with image analysis. So when you run across a problem that you haven't dealt with before, what tools or resources do you reach out to? Do you reach out to other cores? Do you reach out to other people who've handled things? I know with having a mixture of industry and academia here on the call, it's going to be the answer to this is going to be a little bit different.
Visiopharm have training hours, so that's what I tend to do.
Yes.
That comes
Yes.
with the subscription, and and I learn something every time too, and learn a different part of the software. So
I know you're recording, Christina, but I have no issue saying this. I have no problem sending people to Amaris when it's a better tool for a
Yes.
specific application, or if I know of something else that is going to be way better. Because there really isn't like a panacea of everything out there. That's why all these different applications exist for all their sort of niche things. So really it's about trying to figure out what the endpoint is. And sometimes I'll be like, hey, this is how we do things. There are other avenues out there. You might want to talk to, and I'll usually just name drop a core director that I know that has Amaris, like University of Montreal. I'll be like, talk to Chloe about Amaris or something. And I kind of wash my hands of it. But I have no qualms putting people in the right direction. Sometimes with Halo or Visiopharm, obviously, like we can do things on histopathological data. We can do things using pixel classifier and cellpose that are great, multiplex image analysis. But if someone's like, I have spatial transcriptomic data from a Xenium system, I'm just like, don't use Avia. Don't use
No.
Amaris either.
Yep.
They can't even touch that stuff. Use their software. That's what they made it for. And like, why are you trying to use our software? And then sometimes they'll be like, well, because Xenium doesn't do X, Y, Z. I'm like, oh, okay, well, let's I can, we could try to figure out a way to try to put the two together, but there's not going to be any sort of magic bullet when it comes to that kind of stuff.
And I think it's interesting that you bring up the spatial transcriptomics space because I know that's a space that more of us are edging into as these techniques become more broadly adopted and better developed and supported too. And fundamentally, the data types are very, very different, right? So our analysis packages are meant for imaging data. The transcript localization data that comes off those platforms is often graphical format. And so converting a graphical format image and overlaying it onto an actual image is a challenge that none of these platforms were designed to handle, right? And so that's where finding the right tool for the right job is so important. So I think the question I was going to ask next was about emerging tools or workflows that are going to transform how we do image analysis, right? So I think we can all point to AI and AI-driven denoising, segmentation, and other approaches as something that has emerged and become more popular. But are there specific areas where you think these sorts of tools are going to be applied more frequently over the next few years? What's coming in image analysis, or what are some trends that you potentially see? Are people seeing things that have changed, or is it still kind of your standard typical projects that you're seeing on the day-to-day?
I can comment on this. So I think we can start even in the workflow 1 or 2 steps earlier, because this is where I see the most transition for now. Setting, for example, the right setting on the scopes, so getting no saturation or no under saturation. So there are AI tools now built in all the platforms where it's just a button, and I can tell you, maybe I can do a better job, but my users can't. So I don't see under and oversaturation so often since this tool emerges. And we can even think about 1 step earlier when we talked about for the stuff from the beginning, where we say the sample preparation gets important. How often I still get it's a FITSI floor for, and we say, why you do this? And when they start to ask large language models, this gets even sorted out. So because nobody will do this. So I can see that AI and a lot of these new tools even helps us before, because the mistakes we see get less and less and less.
So to build on that, my next question is, what's harder to teach, how AI works or when not to use it? Right? And I don't know that we may not have an answer.
Not to use it. I mean, you get a good feel for it, right? When you see something, if something's too messy, like there's a certain level of messiness that you know that the AI won't be able to pull apart, right? I think that I always tell people try it, right? You never know. Sometimes it's magic, and you press play, you segment, and you go la, it's amazing. But other times you'll bang your head against it for weeks and weeks. So I would always say try it though. I think it doesn't hurt to give some of these things a go. And also, whilst you're trying these things, you're learning the techniques and you're understanding more about how the software works.
Yeah, the confounding sort of principles for a lot of what we do is time and money, right? If we had infinite time and infinite money, we could do infinite science and the world would be a magical place, but unfortunately it doesn't exist. So I think where a lot of these tools can help is saving time and saving money potentially. I think it's far harder to teach people how they work. I don't think that's, unless they want to go into making those tools, I don't think that is a really fruitful endeavor. I think you should know how to use them, right? And that's like me teaching my parents how to use Gemini, not teaching them how Gemini works, right? Like there's a very big difference between the two. And it's useful. One is useful, one is fine, but not quite as useful. Back to the other question you asked of sort of emerging tools, virtual staining, I think has immense potential for saving time and money in first and foremost preclinical and sort of pharmaceutical industry, but even trickling down to what you do at a core. If you can have a really set sort of protocol for like 3-channel fluorescence And whether you're using freeware or commercial solution to say, hey, here are 8 more channels. We didn't have to stain for them or image for them, but you might want to check on this one or this one because it looks interesting, right? And then go back and investigate that. And so save so much time and money in sort of developing a panel of 8 channels where 6 of them are kind of garbage or kind of redundant, and 2 of them are really showing promise. And you can really focus on those. So I think embracing sort of these virtual staining things, I know a lot of people will stand on their soapbox and be like, it's fake data. And yes, it is fake data, but not that you should use it for your research, but to sort of point you in the right direction, I think can save you a lot of time. And I think that's a way that you as a core can sort of hold people's hands and help them see the value in using sort of your expertise, because they probably wouldn't have used that tool or had access to that tool. Let's be honest, most of these tools are really going to be commercially viable and easy to use in the near future. That's going to be a huge sort of value add at the core.
I like that perspective. I think virtual staining is something that just gives me the shivers in general when we start to talk about it. I will fully admit that. And I also think that my challenges with it come from the fact that it's going to be based off of known data, right? In general. And so it goes back to the eyeballs problem, the data set and the input training data that went into developing the model that's then applied to do the virtual staining, right? Are you going to fundamentally make discoveries using a virtual staining platform has always been my question. And my perspective on it, which may be uninformed, is generally no, but I like the idea of hypothesis generation potentially, or at least pointing the research in the right direction. But do other people have thoughts on that?
I have thoughts on Hyplex analysis and sample prep.
Please.
I think I can keep going about these things, but I've seen multiple data sets now, and I never see them done very well unless they come out from the company. Right. So the first thing with the Hyplex data sets is, what are you doing? What question are you answering? Can you use all those channels? What are we doing? What is the question? Right. That's the first thing. The sample prep is another issue. Do I actually believe that you can unmix 30 fluorophores? I'm still on the fence a little bit about it. And then my last point is sample size, right? We're throwing all our eggs in one basket when it comes to these techniques because they're so expensive, and you're only analyzing one piece of tissue coming from one donor that is unknown of any sort of clinical information. So again, making sure that you're not just going with this because it's cool, but you're actually trying to answer a question and you have the right resources. There's my soapbox.
No, I love it. I think it's important for all of us to hear kind of people's perspectives on these different and emerging tools, right? And so the more that we play with them, and all of us have access to different data types and different data sets, all of us have a different knowledge base, right? And I'm with you on the fact that those truly highly multiplexed experiments have to be controlled so carefully, and often for cost reasons, they are not. Right.
Yep.
And that's where it becomes a question of, do you need to push into that level of complexity of science to answer the questions that you're trying to answer? And I think that's a really good point. I know we're coming up on the end here, and we've covered an awful lot in this discussion. I wanted to ask everybody if you could automate one part of the workflow from sample preparation to image analysis, what would you automate? Oh, yes, it looks like you have a thought, Carolyn. Yeah.
Artifact removal.
Oh, that's a good one.
That would save me so much time.
Yeah, I like it.
Rips, folds, things like that.
I think my somewhat basic answer would actually be on the metadata side and file naming, and ensuring that people are following a file naming convention would save me so much time when it comes to data set batching and trying to run analysis pipelines. But does anybody have any other thoughts? Yeah.
General organization as well. Remember that, we're not just looking at one image. Like sometimes we're looking at 80, 100, 160 images. So keeping everything organized and your file structure on the back end so that you and future friends of you will understand what you did is really important too. Keeping your analysis pipelines in an organized fashion, making sure that you have dates and times and understanding in those folder names too. Yeah.
I want to create some type of model. I'm going to call it Better Dappy. When you see it in Nature Methods, you'll know I contributed. It is a generalist, multimodal, multi-magnification background deep learning based enhancement of suboptimal DAPI signal, whether it be extreme heterogeneity, the presence of too many out of focus nuclei, whether it be things that are way too close together. It's going to be automated. Don't worry about it. This is my pipe dream, but very often across all different applications, dev bio, pathology, neuroscience, and just anything, live tissue, fixed tissue. There's always this time in your life where you're just like, yeah, the DAPI's not good on this one, right? And so I don't think anyone ever in science has ever been like,
so
I've never had
sad.
an issue with DAPI.
Yeah.
I've never had an issue with it. No, everyone's always had some issue with it. And so I think it's a big crutch. And it's so prevalent in so many areas of research. It's almost like if you were to invent something that could replace it and be just much more stable or friendly to life and easier to image with a much narrower spectra, you could probably win like a Nobel Prize, honestly, because it's such an underpinning in it. In certain areas like spatial biology, I feel like you hit a wall based on just how good your DAPI is. It's used for the registration of multiple imaging passes. It's used for basically everything, determining the total size of the tissue. It's used for identifying where a cell starts, and it's kind of like groundwork when you're tracking live cells. It's kind of like there, it's what you use, or you're tracking, the hole that it leaves. And so, yeah, that's my ideal is automate improving DAPI.
I
I
am
would add,
100% with you. Sorry
no,
to
you're
interrupt
fine.
you. No, go ahead, please.
No, actually, I was going to add, in addition to DAPI, which yes, we've all had problems with, I would love to automate bubbles removal. Yes, when they come with samples
That's a
and
great
it's
one.
like
Yeah.
so many bubbles, it just it's nightmare.
Agreed.
There's always a bubble that throws off the focus somewhere.
Yes.
Yeah. Yes. No, Daniel, when you mentioned DAPI initially, I was going to ask you if it was just simply pseudo color removal to turn everybody's DAPI's images into monochrome, which generally helps improve things a surprising amount. But I do have to admit that I tell users that their DAPI is bad probably about once a week. So it's a huge problem for sure.
Or they've used it inside the mounting media too, instead of using it separately.
Yes.
Right? So I'll just say that out loud for everybody. Don't use the mounting media that has DAPI in it. Make sure that you do DAPI separately before you mount.
I think we need a BINA sponsored campaign to get Thermo and others to remove those products from there.
Hey, stop it.
Turning into a DAPI truther now. Jeez, wow.
We could have a whole other conversation about DAPI and why it's bad, right?
Yeah.
And probably spend an hour on that in and of itself. But well, thank you all for joining us today. We've got about 3 minutes left. If there's any pressing questions that everybody would like to ask the panel, feel free to drop them in the chat. I think we'll probably close on about, kind of favorite tools and just going back to that. And if you had one image analysis tool that was the only tool that you could use, what would it be? I know it's a terrible question, right? It's a tough one. I don't know. Do people like denoising, for example? There's some people, deconvolution has come up a lot. Are different algorithms and approaches?
I mean, it has to be the neural net for me.
Yeah,
It does. I mean, just like you remember the times when we couldn't segment DAP with nuclei?
right.
It was just frustrating. You try and do it and try and do it and no, and then you run these algorithms and you're like, oh. So I think it has to be the neural net. But you still need to use it in with other tools, right? The pre-processing and the post-processing tools. So I think it all goes together as one. I really do. And using all those using everything in the toolbox to the best ability.
Absolutely. I was waiting for someone to chime in with smoothing filter, right? I mean, there's plenty of things that we could add to our toolkit, right? Yeah. Yeah, for sure. For sure. Well, we've got this amazing Padlet. Everybody's been super active over there. Everyone's been active in the chat, and it sounds like we have plenty more image analysis topics to consider. So thank you all for your contributions. And hopefully we will see you all again on another one of these topics. And I think if there's interest in more specific image analysis topics that are more isolated, let us know. Some of those are in the chat. And we'll be happy to have this panel back and add others to it to discuss those topics. And if we want to have one solely on DAPI, we can absolutely do that. So,
Thank you,
thank you,
everybody.
everybody.